All right. Hello and welcome, everyone. I know there's still a few people signing in here, but I think we'll just get started for today's session. So we have a really exciting day today. We're going to do a Vevo Ask the Experts on Oncology Imaging. So this is really your opportunity to chat with some of our product and application specialists who can help you with any questions you might have about your cancer imaging. I'll be moderating today's session, and we'll also have a brief presentation by one of our product managers, Drew Heinmiller, and I will introduce our panel after a few housekeeping notes. So a couple notes about today's webinar. We are recording the webinar today, so if you weren't able to attend and you want to see some of the Q&A and discussion that transpired, we will be making that recording available. We are muting all the lines throughout the webinar, but please use the Q&A button at the bottom of your Zoom panel at any point to ask any of the questions you want us to discuss on the panel. The questions will be answered during our panel discussion. We do expect this session to be about 45 minutes in length. So with that, those are all my notes for today, and I'd like to invite all of our panelists to come on screen now so we can do some brief introductions. So to start, again, my name is Kristiina Aasa. I'm a scientific application specialist here at FUJIFILM VisualSonics, based out of our Toronto office. Just a random fact about me, I come from a developmental biology background, so I have a really strong love for imaging of the placenta. And with that, I'd like to introduce our first panelist, Stephen Buttars.
Thanks, Kristiina. I'm Stephen Buttars. I'm one of the product managers here at FUJIFILM VisualSonics. I'm here in the Toronto office today. Been with the company for quite a few years and done lots of, had the opportunity to do lots of imaging over the years. One of my favorite tricks for cancer imaging specifically is to do an IP injection of a few mils of saline into the subject animal, because it makes all of the internal organs all float away from each other, and you get really, really nice, pretty images of all the internal organs. And I'll pass it on to Melissa.
All right, well, thanks, Stephen. Hi, everyone. My name is Melissa. I'm also a scientific application specialist with FUJIFILM VisualSonics, also based out of Toronto, our headquarter office. I've actually have quite a lot of experience using the Vevo platform for oncology applications. I've done a lot of research in looking at cancer angiogenesis and changes related to vascularity using the platform. So needless to say, one of my favorite techniques using the system is to use contrast ultrasound imaging to look at changes in tumor perfusion. And with that, I'll pass it to Dieter.
Thank you, Melissa. My name is Dieter Fuchs. I'm joining you from Austria today. Before joining VisualSonics for over 11 years ago, I did my PhD and postdoc in oncology, specifically in neuroblastoma in mice. And based on this, my preferred application for Vevo in oncology is high throughput screening for detection of tumors. And that I'll hand over to Drew Heinmiller.
Thanks, Dieter. It's Drew Heinmiller here. I'm also in the Toronto office. A nice, bright, sunny morning here in Toronto. Leaves are all coming out, and my allergies are just starting from the tree pollen. So, yeah, I'm a Drew Heinmiller product manager here at FUJIFILM VisualSonics. And I would say my favorite application, and I would probably say innovative and probably underpublished application, is looking at hypoxia in tumors with photoacoustic imaging. So you get the high resolution at depth. And that's what I'm going to sort of introduce a little bit today, just about the technology. I'm sure a lot of you are already familiar with our technology, but I do want to just give a really quick, maybe 10-15 minute presentation on a little bit about our technology, the workflow for our technology in cancer imaging, and then just present a couple of publications recent publications that I've found particularly interesting. I want to start off though with a poll. So Mandy, if you can bring up that first poll question, and this relates to really whole body optical imaging versus photoacoustic imaging for oncology research. So the question here is, which is better for whole body imaging in oncology, optical imaging, that's bioluminescence and fluorescence imaging, or photoacoustic imaging? So I just want to take a poll, take a few seconds. I've only given you the two options there, so so click away, and I'll sort of get into it a little later but I just want to sort of start out with that. I know whole body optical imaging is a very, very common tool in oncology research, in vivo imaging, of course. A lot of people using it for looking at things like metastases, tumor burden, and so on. And if you're not familiar with photoacoustics, I'd really welcome you to join the Q&A. Stick around for the Q&A. Queue up some questions. You can enter those at any point and ask away. This weekend I just attended. We're still in sort of lockdown here in Toronto, so I attended this weekend an online beer and cheese pairing night, and one great thing about it is the other attendees were sort of entering their comments and their questions in the panel, and it made it a really interactive, nice experience. So I invite you to do the same. I'm going to end the poll there. And let's share the results. So it's almost an even split, 41 versus 40 versus 60% essentially. The slight preference going to photoacoustics. Now, Mandy, I want to run this poll again after my presentation and just see if those numbers have shifted at all. So I'm going to close that up. So Vevo for Oncology Imaging. If you are familiar with us here at VisualSonics, you'll know that cardiovascular imaging is really one of the really, really strong application areas where we're able to look at heart function in 1D, 2D, 3D, and now 4D, and also looking at other parameters like blood flow velocity and strain and vascular and cardiovascular strain and so on. I'm going to be focusing today on the cancer applications, but there are other applications for our technology in neuroimaging, developmental biology, and a whole host of other applications, whether it be just procedural things like image-guided injections, or looking at smaller animal models like zebrafish, or large animal models. What you're seeing down at the bottom left of that other category is a pig spinal cord. So lots of applications there. Again, I'm going to be focusing on cancer, all different cancer types. Ultrasound is great for soft tissue, brain, kidney, prostate, colon. You can see the list here. Lung is also another one. This is from a nice publication actually showing lung tumor imaging. Not traditionally something that ultrasound is known for, but something that also some of our customers have discovered is good for triaging some of these COVID models that are coming out. Lots of other models here, too many to really list. And it would be interesting to hear from you actually which cancer models you're working on and which you think are the most relevant today in cancer biology. So I'm going to go through Vevo for Oncology, and really, using our system, it can be a real solution from sort of soup to nuts of the oncology workflow. So first thing, you have to induce a tumor model in the living animal, and that can be done via image-guided injection. What you're seeing here is image-guided injection. This is the needle here. There's a couple reflections over here, but this is an injection into the pancreas. So this is all pancreatic tissue here. You can visualize that. You can actually visualize the injection, so you know where precisely you are putting your cells in your tumor models. Next is screening. So you've injected your cells. You want to see, okay, have those tumors, taken up in the organ of interest. So this is great. Ultrasound, high frequency ultrasound can be a great screening tool where we can see very, very small lesions in different tissues. In this case, I believe it's a breast tissue, but all sorts of other tissues there. So you've induced your tumor model. You've now screened for those tumors to see pre-palpable lesions, sub-millimeter lesions, to really see which tumor models are progressing. And then there's sort of two paths. You can watch how the disease progresses in your control group, and then monitor your treatment in your treated group. The most obvious thing here, and I'm sure something all of you are doing, is volumetric. So just looking at sizing the tumor. So we can do that because we can visualize the tumor, we can visualize the tissues with high resolution and get 3D scans and generate those tumor volumes. And then the next level down is sort of looking at more functional information like hemodynamics. So three things here: hypoxia, vascularity, and perfusion. Looking at vascularity first, we're using color Doppler here to assess blood flow in different organs. This is just a sub-Q tumor here. We're looking at a % vascularity measurement, and even cerebral blood flow in this case in a mouse brain with a glioma. So looking at the blood flow there. Going down to the next level is being able to use microbubbles to determine perfusion. So now we're sensitive down to the capillary level, and you can really assess more, slower flowing vessels and really assess perfusion in a tumor. And that's what you're seeing here in this sub-Q tumor model, how that tumor is being perfused. And also having the ability to do, a more in-depth analysis with our Vevo CQ software, looking at peak enhancement, time to peak, wash-in rate, has to do with relative blood volume and blood flow velocity. Hypoxia, or if you remember, this is one of my favorite applications, and I think underpublished. There's plenty of rich soil for planting your experimental seeds and having those show up in publications, because with photoacoustic imaging we can actually see the oxygen saturation status of the blood in the tumor at depth with high resolution because of those optical properties. So we can see well perfused areas with high oxygen saturation, and then hypoxic areas as well, and being able to do that in real time. Finally, we're looking at pharmacokinetics and molecular data. This is where we can use photoacoustics and exogenous contrast agents like dyes, nanoparticles, and so on, and even fluorophores to really characterize these contrast agents in a phantom in this case, using our system, and then going in vivo and looking at basically expression of different molecules through these molecular imaging techniques. Keep in mind, this is all a longitudinal study workflow. So we're really, looking right from the beginning where, we're going from the induction of the tumor model, screening it, then measuring all the parameters of interest, volumes, hemodynamics, and potential molecular information there. And this can be done longitudinally. So you can use the same animal as its own control and go through and have really, really powerful data at the end of it. I want to talk a little bit more about this pharmacokinetics piece. Because of a recent innovation we've had here at FUJIFILM VisualSonics. This is whole body imaging. So whole body ultrasound, and in this case, photoacoustic imaging, where we're able to capture the whole body of a mouse in a 3D scan, and then look at not just the anatomical features, but also molecular features with photoacoustic imaging. So that's, in this case, looking at a contrast agent. I'm going to talk about this in a minute, but a contrast agent that collects in the kidneys, and we're able to visualize that. So this is really looking at that sort of pharmacokinetic model. Now, how were we able to capture images like this? Well, this is with our brand new Vevo whole body imaging setup. So this is basically a new platform. It fits on to the animal imaging station here instead of the animal platform that you would normally use. You clip this on. It's got physiology monitoring, hooks up to our platform here, and an extended 3D motor to really get that whole body of the mouse there. Of course, it's got anesthesia delivery there as well. And you'll notice that we're really, part of the innovation here is actually being able to deliver the light in a different way to generate those photoacoustic signals much more deeply in the animal and the tissue of the animal. So we're actually doing what's called trans and epi illumination. So we're illuminating from both sides and then collecting that data with our ultrasound transducer. And this just gives a more uniform picture of the animal and of the photoacoustic signal within the animal. It just really leads to a facilitated workflow if you're doing these, especially these repeat whole body scans. Now, I come back to the question around which one's better, optical imaging or photoacoustic imaging. So here's a fluorescent image of a contrast agent in the kidneys of a mouse here, whole body fluorescent image. This is great. Many of you are probably familiar with something like this. Now compare it to what we can do with photoacoustic imaging with the co-registered ultrasound. And I'll remind you that, we're able really to, this is a cross section here. So we're going a cross section as it sort of scans through the kidney area. So I'll remind you that, we're getting really good resolution here, sub 100 micron resolution. And we're able to see that biodistribution in those tissues, but we're also able to put it in the context of other anatomy, other, potential diseases, looking at maybe, lymph nodes and that kind of thing. So, the advantages here, I think are actually quite obvious just in this image where, we're getting the whole picture through the whole animal with high resolution and at depth. That image you just saw was acquired using this contrast agent. It's a photoacoustic contrast agent and fluorescent contrast agent called AngioStamp. This is made by a group in Optimal, Grenoble, and it's commercially available. It's targeted to the alpha V beta 3 integrin, which is overexpressed in several tumor types. It's actually, I was looking at a website of a fluorescent imaging system manufacturer, and they've got a whole panel. And this is one of the fluorescent agents they have on the panel that targets this alpha V beta 3 integrin. The difference between theirs and this one is that this one is really truly dual modality, and that it gives a very strong photoacoustic signal and can be used for those kinds of studies. A study just like this one that was published out of that same group from Optimal Grenoble, looking at metastases in the liver, and looking at the uptake of this AngioStamp contrast agent in the liver. Not only that, but also looking at oxygen saturation in the liver as these metastases progress and grow in the liver. How does that change the hemoglobin content and the SO2, the oxygen saturation? And I'll just move on. This is from the same paper. And again, we've got another comparison here between whole body optical, both bioluminescence to look at, those tumor cells are bioluminescing, and fluorescence as well, looking at the AngioStamp with fluorescence. I'll invite you to just take a look at these images, which I'm sure you're familiar with, and then take a look at these images, which are actually cross sections through that section of the animal, looking at the liver in this case, and being able to assess not just the extent of AngioStamp that has gathered there, but where has it gathered, being able to quantify it accurately, and also being able to look at just other morphology of the liver and things like oxygen saturation. So you really get sort of three things at once. You get your oxygen saturation, your anatomy, and your signal here from your fluorescent or photoacoustic agent. And then if you wanted to, all of the other parameters like blood flow, perfusion, that kind of thing. So, I'll just ask here, no, I'm going to keep going before I have Mandy relaunch that poll. I'm almost done here. Just another couple of publications. This is a really nice one out of UPenn by Dr. Siegel's group here, looking at, in this case, hepatocellular carcinoma, looking at treatment with therapeutic ultrasound to treat vasculature. So what they do is, they have these HCC tumors growing. They want to disrupt that vasculature. So often they'll use microbubbles, and they'll disrupt those microbubbles with ultrasound, therapeutic ultrasound, and that will destroy the vasculature. This is a way of monitoring that therapy using Doppler to look at the blood flow in those tumors, and also using photoacoustics to look at the hemoglobin content, or relative blood volume really, and the oxygen saturation, how much oxygen that blood is carrying. So in all cases, you see these effects of these therapeutic doses. So, this is now a multi-parametric assessment of the effectiveness of that treatment. So nice paper here. This is a really nice one as well, where instead of just treating a tumor here, they're actually using an alpha V beta 3 integrin targeting agent. Remember, that's just like the angiostamp agent. They used a different one, an IR820 based agent in this case, where they would grow these, again, HCC tumors, grow the tumors, inject this alpha V beta 3 integrin targeting agent, and then they would go in and use this for guidance for surgical resection. So they basically look for the glowing tissue and then resect it and then image again to see if they got the whole tumor. And as you can see, and as they pointed out, actually, once they started the surgery, it was actually a little bit tricky just with the ultrasound alone to determine the tumor boundaries in the liver. And they weren't sure, how much healthy liver are we taking, or are we even getting the whole tumor? The boundaries between those two tissue types were a little bit harder to see. This is where photoacoustic imaging came in. And because of this alpha V beta 3 integrin binding of the agent, it allowed them to visualize the actual tumor tissue and remove that tumor tissue. So this was a really interesting study, obvious translational applications here as well. This is another part of the paper which I really liked. That was obviously preclinical data. They were doing that in mice. Here, they actually took patient specimens from the clinic. They stained them with this contrast agent. They imaged them in 3D with photoacoustic imaging, and then they just looked, okay, where, which parts are being stained. And they found a really, really good correlation between the tumor and the contrast agent. So the contrast agent was sticking to the tumor, not to the normal liver tissue. And they got, there's a lot of images here. I wish I just picked out one or two, but you can go through it and maybe appreciate they've co-registered a photograph with their 3D photoacoustic and ultrasound scan with their H&E stained sections. And they have really beautiful correlation between these three. So again, the translational impact of this is I think really obvious here. Finally, this is my last slide here. This is a really nice paper looking at reporter imaging. So reporter imaging, and I'll come back to the optical thing for a second. Reporter imaging is used, to label your cells. So your bioluminescence, you're looking at your luciferase enzyme. So you've transfected your cells, you inject the luciferin, and the luciferase is being expressed by those cells, and you have those tumor cells lighting up. Well, this is a similar approach, but using photoacoustics instead. So they transfected these cells to have a gene that would increase the uptake of a photoacoustic dye, indocyanin green. So these cells, these tumor cells, now were more prone to be able to take up ICG. So they grew these tumors, they injected ICG systemically, and sure enough, they saw a greater uptake, 2.3-fold uptake in those tumor cells. So this is really analogous to that luciferase assay approach. The difference here is that now, instead of having these images up here, just sort of blobs on the surface of an animal, these are, again, cross sections where we can say where exactly those tumors are, what is the morphology of the tissue around it, and really go in and get that resolution at depth. So before we launch into the Q&A, Mandy, is it possible to relaunch that first poll question? I'm wondering how convincing I've been in convincing the people attending that, which one's better, optical or photoacoustics. And if you're still saying optical, I'd love to hear why. We've got a Q&A session that's coming up just now. I'd love to hear your answers about this and why that tool is useful to you. But right now, it really looks like we've done a nice job of sort of shifting the ground a little bit here. I'm just watching some of those answers come in. I'll end the polling here because I don't want to take up too much more time. I want to save some time for questions, obviously. And as you can see here, the results are now pretty overwhelmingly in the favor of photoacoustics. So we've had quite a big shift there, a 15% shift, toward sorry, a 25% shift over toward the photoacoustic side of things. And frankly, this is no surprise. There are obvious advantages to using photoacoustic imaging for doing some of these assays. And if you have any questions about this, we'd love to hear about it. And with that, I believe I will move on to the Q&A portion and hand it back to you, Kristiina.
Thank you so much, Drew. And I certainly hope that presentation kind of spurred some interest and some questions amongst you and about how you're currently performing your cancer imaging. So I'd like to please encourage you to use the Q&A button. There are no silly questions. All questions are welcome. At this point, I'd like to invite the rest of our panel to come back on video here. We do have a really great expert panel of individuals who can answer some of your burning oncology imaging questions. So I'd like to actually start, if Mandy, you wouldn't mind launching that one more poll, just to get an idea of the audience we have participating today and what you're currently using with your imaging platform. So what are you currently doing with your imaging platform? A couple options there. Oxygen saturation, you might win Drew's heart with that. As, he alluded to that's his favorite technique here. Are you looking at perfusion vascularity? So you'll really be speaking to Melissa and her love of bubbles and perfusion kinetics. Are you looking at tumor vasculature? Are you doing image-guided injections or theranostic molecular imaging? Are you after Dieter's heart and doing high throughput screening? Or currently, are you not using actually a Vevo system and you're here to just really learn a little bit more about what our technology can offer you? So we have a lot of people answering here, and thank you so much. I'll give it 20 more seconds just for the last few people, and you can select multiple options here. So select all of the ones that are applicable to you, and it'd be great to see what everyone's dealing with here. All right, so things have slowed down, so I'm going to end the poll now, actually, and we'll share the results. So we see a lot of people doing tumor volumes. We know we have a lot of publications in that field, so maybe in 3D, maybe in 2D. We do have some people interested in screening. So Dieter, I'm sure you're happy to see that. A little less in tumor oxygenation, and maybe that will change after Drew's intriguing presentation there. So I want to thank you so much for that poll. And at this point, I'd really like to launch into the questions. And again, please don't be shy. We've had a couple of questions coming in, and I'll start with one that came in here that really dovetails nicely with Drew's presentation. And the question is oxyhemo mode better than color or power Doppler? And please, anyone on the panel, jump in here.
I can jump in on that, given the sort of theme here. I mean, it depends what you mean by better. You're really looking at different parameters. There are certain parameters that are kind of overlapping. So, you're looking at blood. So there's going to be an overlap there where the presence of blood is. So for example, maybe things like the total hemoglobin measurement that you can get from photoacoustics, that might correlate with some of that blood flow. On the other hand, if you've got, say, inside a tumor, if you've got hemorrhaging inside a tumor, that's not going to be detected with power Doppler because there's no flow there to detect, but it will be detected with photoacoustic imaging. So it's really this sort of what I see as overlapping, but really complementary data sets that you're going to get using a combination of those parameters. And it's going to tell you more about the vasculature and functionality of that vasculature in the tumor than any one modality alone.
I'd like to add to that. I can only agree with what Drew said. With the Doppler, color power Doppler, you look into established large blood vessels. With contrast enhancement, you look into capillaries, capillary perfusion, capillary function. And then when you switch on oxyhemo, you go for photoacoustic imaging, you really drill it down to the molecular level of how good is the tissue with coping with lack of perfusion, for example. There are studies where you do a sort of oxygen challenge where you give the mouse different level of oxygen to breathe and see how quickly a tissue changes or can adapt to that change, or how long it takes to go back to baseline as opposed to a healthy tissue. So really relevant to look into how good a tumor is doing compared to an adjacent normal tissue.
Great. Thank you both for that. If no one has anything else to add to that, I'll move on to the next question. So still talking about photoacoustics in this sense, can photoacoustics identify sentinel lymph nodes?
I actually had, if I can just quickly share, because I actually had, I saw the question, I sort of, I just kind of saw this. So that's a great question. You know, sentinel lymph nodes, of course, being those nodes that are the first site of metastasis of a metastatic tumor model. And this is from a really nice paper that shows exactly that. So there was a primary tumor in a tongue that it metastasized down to these cervical lymph nodes. And they had a contrast agent. This is made by the group, a custom contrast agent that would actually target those metastatic cells. So they injected this contrast agent at the site of the primary tumor. It flowed down into the lymph nodes, and it would only be detected if it was internalized by those cells. If there was no tumor there or no metastasis there, you wouldn't see the signal from the photoacoustic agent. So it actually changed its optical properties depending on if there was the presence of tumors or not. So that's just one example of being able to detect metastases. I have seen other papers where you're really looking at oxygen saturation changes in the lymph node, and then just morphological changes. You know, what does the lymph node actually look like in high resolution ultrasound? Those generally, of course, come a little later when the metastasis is quite developed, but those are some of the approaches that I've seen.
Yeah, and I'll just add on, I think this really speaks to the multimodal imaging aspects of the platform. And so just as Drew has already alluded, you can do some really nice volumetric imaging with just a high resolution ultrasound. But then on top of that, when we kind of look into more molecular functional level imaging, that's really where sort of the Doppler imaging comes in, where photoacoustics really comes in. So it's really a combination of the different modalities that can really be utilized here.
And also add for sentinel lymph node detection, something that is used in the clinic, for example, to inject different dyes close to a tumor, like, for example, ICG would be one of those dyes. Inject around the tumor, they will drain, and they will drain into one or several adjacent lymph nodes, and then it can be perfectly detected with photoacoustics, knowing precisely where it is, at what depth, and with the high resolution B mode image, you can assess the size of the lymph node, looking collateral and see the contralateral lymph node and see if anything has changed in size.
If I can just share, I don't get to show this too often because it doesn't come up very often, but this is an example of actually dye that was injected in the hind limb of an animal. It's actually flowing through the lymphatic system. So lymphatics are so small that even with high resolution ultrasound, you can't see the lymphatic vessels. You can see lymph nodes, no problem. You can't see the vessels. So this is an example where there's actually two different dyes, I believe Evans blue, and then this sort of liposomal form of ICG. And as you'll see here, when you injected them, they actually have different pharmacokinetics flowing through the lymphatic system. So the blue dye, if you'll see, it sort of flows up here and into the lymph node. This is a popliteal lymph node, and then it flows out. You can see it flow out to the left there. The green stuff, that's the liposomal ICG. It's more chunky and it's bigger. It kind of gets stuck in the lymph node itself. So this is really actually looking at like lymphodynamics in real time with photoacoustic imaging, a really neat tool. And again, maybe more of a niche area, but really intriguing because there really aren't that many in vivo tools to look at lymphatics.
Great, thank you all for that. So the next question, and this follows nicely with, we've been talking a lot about contrast agents and dyes. So this is more of a two-part question. How difficult is it to create contrast agents that are targeted, and what are the limitations? For example, how overexpressed does the antigen need to be? And kind of secondary to that, and I think these flow together, with bioluminescence, you can look at a wide array of markers, so you can really image anything. And the concern here is that photoacoustics, the contrast agent is hard to make, and there's a significant limitation in what you can target. And can anyone speak a little bit more to that? I know that was a bit of a mouthful of combining them together, so I can repeat if needed.
Juju, do you want to start or?
Oh, go ahead.
Okay, I mean, I can kick it off here and then feel free, everyone else on the panel, to add on to this. I think this is really a great question, and we do get this a lot. So there's abundance of work with different groups actually making their own nanoparticles or photoacoustic compatible agents that are targeted, or or making them for therapeutic purposes. So the question of, is there something that's maybe easily adaptable, is something that we actually discuss with our customers quite a bit, our users quite a bit. I'm actually just going to probably bring up a pretty good example. And if I may, I'm going to share my screen here of an example where a group actually used something that is off the shelf to really do this targeting. And this is something that could be looked into. So here, what you're seeing is actually a study that's done coming out of Stanford, where they're actually looking at using photoacoustics to detect aggressive sort of memory tumors. So here they're looking at a very specific biomarker, the B7H3. So if we start from the left of the slide, they did a lot of sort of characterization of the expression of this particular biomarker. Maybe that's kind of goes into that question of how well does that target has to be expressed. I think there's a little bit of research that can be done ahead of time, sort of understanding how overexpressed this biomarker is. So they've taken that from that clinical side and then translated to a mouse model, understanding that this is a upregulated biomarker that can be targeted. And this is where I think where that component of, is there things that's off the shelf that can be used, is the actual dye they're using is a very common dye. It's ICG, indocyanate green. It's actually clinically approved. And here they've actually bought something that's right off the shelf and then attached it to this antibody that they're targeting, leading to some nice characterization photoacoustically, and of course, some in vivo imaging showing that you can use this off-the-shelf package to be able to do that nice tracing of this biomarker in its overexpression in memory tumors. So if there's any interest, this is a particular vendor that sort of supplied this ICG dye that can be easily attached to different targeted biomarkers. And so this is sort of just that great example. And I'm going to stop sharing it right now of how you can use something that's off the shelf to be able to do this. And there's also some other, I think, dyes out there that can be used as well.
Yeah, that's a great paper, Melissa, and they've done a great job of using something that's ICG itself is clinically approved. This conjugatable form is not, but I think this points out that photoacoustics is a relatively new imaging modality, so it's not quite where BLI and FLI is in terms of its maturity. It's still growing, and I think the growth of photoacoustics is actually going to be in parallel or bolstered by the development of these contrast agents. Many of our customers are doing work right now on developing new contrast agents for therapy, diagnostics, a whole bunch of different things. So I would agree that, yeah, you can image a lot more. There's these whole panels of fluorescent agents for looking at a variety of markers that will come for photoacoustics. It's just a little earlier on in the development stage.
And definitely lots of our users that are out there are also developing unique ultrasound contrast agents, microbubble-based agents. So the most common, well-known agents that Melissa spoke about and some of the work that she's done and we've all worked with are phospholipid shell microbubble based, but there are lots of other different approaches for doing ultrasound contrast agents, different types of lipids, hard shell, soft shell. So once you start looking into contrast agents, whether it's on the ultrasound side or the photoacoustic side, there's a huge range of possibilities out there, targeted and untargeted both.
And I'd like to add, for preparing different contrast agents, as Drew had in as you had in the presentation, the in vitro screening, allowing you to really assess proper photoacoustic properties, looking into storage conditions, dilution series, signal to noise. It's really, you can do all of that, all of the characterization and creation of new photoacoustic contrast agents without using a single animal. So a lot of preparation can be done before going in vivo, which really helps in creating and doing all the base work and the groundwork.
Great, thank you so much. So the next question is about lung imaging. So are you able to do lung imaging using high frequency ultrasound photoacoustics, or can you speak a little bit more about how that works and if you're able to do things like measure granulomas in the lung? Don't be shy here.
I was just thinking to myself, I was wondering if I have any lung tumor images in my image bank, but I don't. There's definitely been some lung work. We actually had a webinar not that long ago from a group that was doing lung imaging. Now they were more specifically, it was actually specifically a COVID-19 related research project, but there's definitely been more of a focus and interest in lung imaging lately. Lung tumor imaging has definitely popped up from time to time. I do recall seeing some images one time, some time ago, that were quite nice of lung tumors, especially as they got a little closer to the chest wall, you could really see them, but I definitely don't have any of them in my image bank. I don't know if anyone else does.
There was a group in France that actually developed a model, lung tumor model, specifically for ultrasound and photoacoustic imaging. So they, this model, so normally the lung looks like this bright, big, thick, bright line because it's filled with air and the ultrasound doesn't go through the air. So you get this really thick, bright line. Whenever you have any anomalies in the lung, if you got, it's filling up fluid, for example, or you have tumors growing, as long as there's no air kind of above from where you're imaging, it shows up beautifully because the contrast between the normal lung and this is huge. And that's what this group did in France. They grew tumor models specifically at the periphery of the lung so that they could be visualized and they could see the growth and oxygen saturation in those tumors.
I have the paper ready if you'd like me to show Drew.
Sure,
yeah. And as you're about to share that, dear, we'll put in the chat a link to a publications PDF. So we have some key publications that we put together in the field of oncology imaging. And one of those sections is on lung imaging. So if you want to check that out and follow that link, you can see 3 references to papers that are really nice papers when it comes to lung tumor imaging.
Yeah, so this is the paper that I mentioned, the group in France from Orléans, creating an orthotopic model for lung tumors, being able to detect the tumor with ultrasound, adding photoacoustics to look into tumor hypoxia, how the tumor develops, even though it's nicely growing in the lung, it has a very hypoxic area, and then also looking into the volume of total tumor versus hypoxic region or hypoxic volume inside the tumor, and linking it later on also to molecular image with help of molecular imaging, ultrasound molecular imaging, looking specifically for VEGF receptor 2, so angiogenesis. So tumor monitoring, sizing, volumetric quantification, looking into the angiogenic region. You see it's very much to the center where you also have the hypoxic region through multimodal imaging using the Vevo platform. And interestingly, if you look into conventionally, CT is used nicely on lung tumors. And in that, with this model, there's a nice linear correlation when you look into volumetric quantification using ultrasound in vivo and the CT. So it makes a good read, and you will find this paper in the bibliography on the VisualSonics webpage.
Yeah, and I'll just add to this. There's actually been a pretty recent Jove protocols paper that was published actually looking at detection of lung metastasis. Sort of going back to what Drew was mentioning earlier is if you look at the normal lung, we're actually seeing these A-line artifacts, but when there's these changes, that's creating sort of tissues that now that the ultrasound can actually reflect off of, that actually changes that image. And that protocols paper actually does a pretty good really introduction to how that change could be evaluated. So as Kristiina said, we are happy to share some of those papers with you on the list of publications that we will provide.
Yeah, and that both those papers, the Jove paper and Florian Race's paper that Dieter just shared, are both in that publications PDF that we've put in the chat if you want to check that out. Excellent. So we had some nice discussion about lung imaging here. I think that goes right into our next poll question quite nicely, where again, we want to try and gauge the rest of our discussion here based on some of everyone who's attending in our audience and what kind of imaging you're doing, what cancer model specifically you're working with. So if you wouldn't mind, again, this is a multiple choice question, so you can have multiple answers. So what types of cancer models are you working with? There's some options there. Is it pancreatic, mammary, kidney, bladder, brain, or other? I assume we're going to have at least one other because we did have that lung imaging question there. So yeah, if you wouldn't mind putting it in our chat there, or sorry, putting in the answer to the poll. Again, a few of those at least are covered in that key publications PDF. But if you ever have questions or want to get access to more of our publications, we actually have a very thorough publications database on our website. You can look at it from our main website. You can go under resources and publications library. So there you're able to filter out by oncology and actually see all the publications that have come out using our system in that particular field. So I encourage you to check that out if you have any further questions or want to do a bit more lit review on it. Helps narrow things down a bit more than doing a generic PubMed search. All right, we have some nice response here, so I'm going to end the poll. We really have, it's a mixed bag. Kidney, is in the lead, 64% of the votes, but it really is a nice spread. So it's nice to see that we have a good representative of all these different fields within oncology being participating in today's session. Perfect. So I'm going to stop that share. We're going to continue with our questions again. Please use the Q&A button at the bottom of your Zoom panel to ask any questions. So the next one is really about frequency. So if the transducer, or does the transducer frequency, for example, an MX400 versus an MX201, does it influence the resolution of the photoacoustic image?
I saw that one coming and I queued up a couple of things to show here. So
Yeah,
I'll take that one. The best way to answer that actually is to have a look at our website. So if you go to our website, visualsonics.com, and if you log in, go ahead to the login page. You can log in, or if you don't have an account, you can create one right there on the website. Once you get into that section of the website, you'll find a whole suite of various documents that are not available on the public site, including things like spec sheets. So if you, I happen to have the Vevo 3100 specification sheet queued up here. And if you look on the second page of that, you'll get a breakdown of the different transducers and the different attributes of those transducers. And when it comes to the resolution question, there's a couple of different resolution columns here, but the axial resolution is really one of the key ones to look at. And you'll see fairly quickly as you start to compare that as you go up or down in frequency of the transducer, the resolution of the image, the resolution that you're able to attain with that transducer also goes up or down. So as an example, the highest resolution transducer that we have, the MX700, will give you an axial resolution of 30 microns, versus down at the lower end, the 15 megahertz center frequency, will actually give you an axial resolution of 100 microns. So there is this sort of opposed relationship, this interactive relationship between the transducer frequency and the resolution. So across the whole range of transducers that we have, you've got this wide range of depths of penetration and resolution capabilities to go from sort of a large field of view to look at whole body, some of the whole body images that Drew showed on the lead in, those are typically done with the lower frequency transducers to get a wider field of view. You do sacrifice a bit of resolution, but some of the screening images, especially that mammary fat pad tumor image, that's typically done at a very high frequency and a high resolution to really see those small structures as they start out. Okay.
Okay, great. Thank you so much, Stephen. So we have about 9 more minutes here, so we'll try and fill this up with engaging discussion because I think I'm having fun. I don't know about everyone else. So as we know, imaging brain tumors are difficult via ultrasound. I think we can all acknowledge that. Is creating a window in the skull the only way to really anatomically image for a tumor? Or do you have any other suggestions or tips and tricks for another approach or a different technique? You can tell it's a little more difficult. Nobody wants to jump in here first.
You've done quite a bit of brain imaging.
Yeah, absolutely. I can sort of start this off. I think maybe the first question is really the model that you're using. With our experiences with general mouse imaging, it is dependent sort of on that thickness of the skull, because we all know sort of ultrasound does not interact with bones very well, so that really attenuates once you hit that first surface layer. From my experiences, I've generally liked to go for a lower frequency transducer to give me that nice penetration depth. I will say, you know, anatomically, if you're really looking at being able to find the tumor and evaluate the volume, then we do, you do want to have a clear image, and this is where sort of cranial windows or even thinning the skull could help. So sometimes even removing the scalp and thinning the skull a little bit can really help with that penetration and also visualization of the lesions. I mean, there's been quite a number of publications out there, even sort of introducing sort of cranial windows that you can be done in sort of for longitudinal imaging so that you can actually be able to evaluate tumor growth over time. But if you're really looking to do sort of really small submillimeter tumors and looking at accurate volume, thinning the skull is and having that is necessary, I will say. Now, with that being said, there's a lot of actual functional imaging that you can done with the full skull intact. So if you're maybe looking at things like, you know, disruptions in blood flow in the brain, or even looking at oxygen saturation in relation to some of those functional changes to the tumor, then, you know, we can actually visualize that quite well with a fully intact skull at that point.
Yeah, Melissa, I know there was a paper of a number of years ago now by a group who is looking at this. I forget what it's called, but it's like PDMS film. It's basically some sort of like polymer that's acoustically transparent and optically transparent, and they were able to implant that in the skull of a mouse, and they claimed you could even inject through it, and it would be fine. You know, you could inject through it and it would still maintain its patency somehow.
Yeah, absolutely. It's this polyethylene cover slip that's used that's kind of glued on. I've actually have a quick slide pulled up. So maybe let me see if I can just cue that on so we can sort of show the audience here what's done. So just give me one quick second.
Yeah, and the difference is really, I mean, they are quite striking. I mean, not surprisingly, you can even go up in frequency if, this is great. You can even go up in frequency if you're able to have that cranial window. You get really nice resolution.
And I'll add that the full citation for this paper is in that publications PDF again, under the brain cancer section.
Yeah, and this group actually did a number of publications. This is just an example. So on the left, you can sort of see how that cranial window works. This is that polyethylene cover slip that we're talking about that you can actually mount to the head of the animal. And this is more a longitudinal imaging experiment. So it does actually pertain quite well over time. But I think the great thing here is if you sort of look at some of the tumor volume changes that they're tracking, we're really looking at very, very small tumors. We're looking at things that's down to 5 millimeters cubed. So you're really able to get that high anatomical resolution to be able to track these millimeter tumors, really. So this is just an example from the group. And again, as Kristiina has mentioned, the full list of publications is also available to you.
Great, thanks so much. We're heading to the
Can
last.
I just show one more thing? I mean,
Yeah,
if people haven't seen some of these images, and sorry, it's going to take me a second or two to bring this up, but if people haven't seen some of these images of the brain specifically, and again, I think this is one of those sort of what the term I've been using is underpublished applications using ultrasound and photoacoustics to look at this. I mean, this is a rat pup with the skull intact. There's, the skull isn't quite fused yet. There's a pulled glass pipette doing an injection into the corpus callosum here. You can actually see this sparkling of, I don't know if you can see my cursor, but I'm pointing in the top right image here. You can see the superior sagittal sinus and blood sparkling there. Looking at oxygen saturation, this is the image on the top left, skull and skin totally intact. And this is an older image. Melissa has much better images than this by now. But then, even just looking at the brain that's been excised. So this is just a brain, the bottom left is the brain in saline, just gorgeous. I mean, absolutely gorgeous anatomy coming through here with a 40 megahertz transducer. So we're looking at 40 micron resolution in this case. And then even looking at blood flow using microbubbles in the bottom right. Both blood flow looking at Doppler, and then on the bottom right, the right-hand image is looking at blood flow. Again, that one, the bottom right image, that's skin and skull intact. And you're seeing these absolutely gorgeous, vasculature coming in. This is a stroke model, actually. So you can see on the right hemisphere there, there's impaired flow in that case. We say, oh, ultrasound can be tricky through the brain. When you're looking at a mouse skull, which is already pretty thin, and looking at some of these tools here, yes, in some cases it's quite ideal situations, especially when you scoop the brain out. But the advantages of having that resolution, having that real-time aspect, I really think this is again ripe for publications looking into this using these tools.
Yeah, and just adding this for brain tumors, just last week we had an oncology workshop where Dr. Calber from London in the UK presented on something on reporter genes, so creating cells, enabling them to express a reporter gene that can be detected with photoacoustics, so you are completely non-invasive and see where the tumor is, how a tumor is spreading. We're working on it's in post-production right now, but there should be a section on the webpage as a video on demand if you're interested in that. So please check it out.
Great, thank you so much. So I'm going to, the final minute, just to really quickly, if someone could comment on how to increase throughput. We had a bunch of people on here who were interested in renal cancer imaging. So if someone has just 30 seconds, they can say on how to increase throughput on detection of kidney tumors.
I like throughput, so I quickly take that one. If the ethical protocol allows, you could work with conscious animals, holding it like you were giving an IP injection, and quickly screen, just scruffing, no clear depilation needed, just a quick trim of the fur, and you have like tumor yes, no. If you want to know more, then you can dig into volumetric quantification. You can use a foot pedal to save the images for later, or use voice control to have the images stored, and then you can do the analysis later. But it really gives you a quick way of screening through the tumors.
Perfect. Thank you so much. So with that, I want to respect everyone's time. I want to thank all of you. I think everyone in the audience can tell that we're all a very passionate group of scientists who work here at VisualSonics, and it's hard to cut us off when we're talking about using our technology to help people and help people image. So with that, I'd like to thank everyone for attending so much. Please check out our website. We have a bunch of different links here. I'm not going to go through this all with respect to time, but we do have upcoming webinars if you'd like to register for them. Please contact us using our web portal if you have any additional questions, or you can contact us on social media. And we do have a learning hub, which is a training portal for training videos. And please check that out. You can find the link on our website. So with that, I'm going to stop sharing so that my colleagues here can all say goodbye. Thanks so much, everyone.
Thanks, everybody.
Thanks, everybody.
Thank you. Bye.
Bye now.
Have a great day.